everyone. Today we will learn about complement fixation test. In general, complement fixation test (CFT) are best performed in the reference laboratories where faculties exist for the careful standardization and control of reagent which these tests are required. And we will also learn how this test came into existence. In 1894, Richard, a German scientist, had discovered that when cholera bacteria were injected in the peritoneum of a guinea pig, immunized against the infection, the pig would rapidly die. This bacteriolysis, Brodet discovered, did not occur when the bacteria was injected into a non-immunized guinea pig, but did so when the same animal received the antiserum from an immunized animal. Moreover, the bacteriolysis did not take place when the bacteria and the antiserum were mixed in the test tube unless fresh antiserum was used. However, when the Brodet heated the antiserum to 55 to 56 degrees centigrade, it lost its power to kill bacteria. Now, introduction. Complement is the protein present in the normal serum. Whole complement system is made up of 9 components, that is from C1 to C9. Complement protein are heat liable and are destroyed by heating at 56 centigrade for 20 to 30 minutes in a process called heat inactivation. Complement binds to antigen-antibody complex. When the antigen is complexed with antibody on surface of cell, complement cause lysis of cell. Components of CST test system. Here are some components. Antigen. It may be any antigen, soluble or particulate. Antibody. Human serum. Paired sera are used to detect recent infections. Complement. It is pooled serum obtained from 5 guinea pigs. It should be fresh or specially preserved as the complement activity is heat liable. The complement activity should be initially standardized before using in the test. Erythrocytes, SHIP RBC act as the antigens. Hemolysis. Rabbit antibody to SHIP red cells prepared by inoculating SHIP erythrocytes into rabbit under standard immunization protocol. Here are the principles of CFT. The principle of the CF test is that antibody present in patient sera when mixed with the corresponding antigen will fix or bind complement. This fixation of complement is determined by using an assay system consisting of SHIP red blood cell centralized with anti-SHIP red blood cell and measuring the percentage of lysis of SHIP red blood cell of the all components has been fixed. The indicator SHIP red blood cells will not be lysed. Now, CF tests are interpreted as follows. If antibody is present, no hemolysis will occur, and if antibody is absent, hemolysis occur. In that, patient's sera should be treated with each of the antigen. Since there is some overlapping of the antigenicity between the various functions of the cell system of the disease are very similar. Higher CF titers are usually observed on patient sera when they are tested against the same antigen as the erythrobiological against their infections. CFT consists of 2 steps. Step number 1 is a known antigen is inactivated. Patient serum are incubated with the standardized limited amount of complement. If the serum consists specific complement activity antibody, the complement will be activated or fixed. by the antigen-antibody complex. However, if there is no antibody in the patient's serum, there will be no formation of antibody-antigen complex, and therefore complement will not be fixed but will remain free. In this slide, we may also see that here you can see that antigen plus complement plus serum with antibody against antigen is present in the test tube. Here in the next, we may see that in this slide, in this test tube, complement fixation occurs. In next, we see that SHIP RBCs are added with antibodies to SHIP RBCs. And in the test tube, we see that no hemolysis occurs, which gives a positive test. All available complement is fixed by the antigen-antibody reaction. No hemolysis occur, so the test is positive for the presence of antibodies. In next, we see that antigen is added with complement without antibody in the serum. In the test tube, next test tube, we see that in the same test tube, no complement fixation occur, and if the RBC of SHIP is added with the antibody, the hemolysis occur, which gives a negative test. No antigen-antibody reaction occurs. The complement remains and the red blood cell are lysed in the indicator stage. So the test is negative. So here we have studied both positive and negative test. Now, as we studied the first step, the second step is detected whether complement has been utilized in the first step or not. In this done by the adding the indicator system. If the complement is fixed in the first step owing to the presence of antibody, there will be no complement left to fix to the indicator system. However, if there is antibody in the patient's serum, there will be no antigen-antibody complex, and therefore complement will be present free or unfixed in the mixture. This unfixed complement will now react with the antibody coated SHIP red blood cell to the bring about the lysis. Thus, no lysis of SHIP red blood cell indicates the presence of antibody in the presence of antibody in the test serum, while lysis of SHIP red blood cell gives or indicates the absence of antibody in the serum. Now, we will see that control should be used along with the test to ensure that antigen and serum are not anti-complementary. The appropriate amount of complement is used. The SHIP red blood cell do not undergo autolysis. Advantage: ability to screen against a large number of viral and bacterial infection at the same time, and this test is economical. Disadvantage: it is non-sensitive test, cannot be used for immunity screening. Second is time consuming and labor intensive. Third is often non-specific. Example, cross reactivity between HSV and VZV. Now, here are some of the modification of complement fixation test. Here we study about the first modification is indirect complement fixation test. In this modification is used when serum which do not fix guinea pig complement is to be tested. Here the test is set up in duplicates. After set 1, standardized anti-serum to antigen where it is not fixed, complement is added to set 1. If antibody were not present in the test serum, then the antigen would be reacted with the standard anti-serum, fixing the complement. On the other hand, the antibodies are present in the test serum and antigen would be utilized in the first step. So, no reaction would occur between the standardized anti-serum and the antigen, and therefore no fixation of complement would cause lysis of SHIP red blood cell. Thus, in this case, hemolysis indicate a positive result. Second is conjugating complement absorption test. Here, Horse complement, which is non-hemolytic, is used. The indicator system used is sensitized red blood cell mixed with bovine serum. Bovine serum consists of beta globulin called conglutine would also combine with this complement, causing augmenting of the red blood cell, indicating a negative test. Here we have seen that in the first fixation test there is positive test, and in the second coagulating complex absorption test it gives negative result. Now the third is immune adherence. When some bacteria combine with the specific antibody in the presence of complement and some particles such as erythrocyte or platelets, they adhere to the erythrocytes or platelets. This is called as immune adherence. Now fourth is immobilization test. In this test here, antigen is incubated with patient's serum in presence of complement. If specific antibody is present, it would immobilize the antigen. Example is Treponema pallidum immobilized test, considered gold standard for the serodiagnosis of syphilis. Now fifth, that is E, is cytolytic test. The incubation of a live bacteria with its specific antibody in the presence of complement lead to the lysis of bacterial cell. This is the basic of biological antibody test used to measure anti-cholera test. This is how modification of complement fixation test is done. Thank you.